DNA is transcription unit in DNA is defined by three regions: a promoter, the structural gene, and a terminator. The promoter is located towards the 5' end of the coding strand. It is a DNA sequence that provides binding site for RNA polymerase. The presence of promoter in a transcription unit defines the template and coding strands. The terminator region, located towards the 3' end of the coding strand, contains a DNA sequence that causes the RNA polymerase to stop transcribing. In eukaryotes, the promoter has 80 rich regions called Tata box, Goldberg Hogness box, and in prokaryotes, this region is called Prebno box. Besides promoter, eukaryotes also require an enhancer. Those two strands of the DNA in the structural gene of a transcription unit have opposite polarity. DNA-dependent RNA polymerase catalyzes the polymerization in only one direction. The strand that has polarity 3' to 5' acts as a template and is called as the template strand. The other strand, which has the polarity 5' to 3' as sequence same as RNA except thymine instead of uracil, and is displaced during transcription. This strand is known as coding strand. The structural gene may be monocistronic eukaryotes or polycistronic prokaryotes. In eukaryotes, each gene transcribes a single mRNA and encodes information for only a single protein and is called monocistronic mRNA. In prokaryotes, clusters of related genes known as operon found next to each other on the chromosome are transcribed together to give a single mRNA and hence are polycistronic. Before starting transcription, RNA polymerase binds to the promoter, a recognition sequence in front of the gene. Bacterial prokaryotic RNA polymerase consists of the core enzyme and the sigma subunit. The core enzyme 2 alpha beta beta 1 and gamma is responsible for RNA synthesis, whereas a sigma subunit is responsible for recognition of the promoter. Promoter sequences vary in different organisms. RNA polymerase opens up to the DNA to form the transcription bubble. The core enzymes moves ahead, manufacturing RNA, leaving the sigma unit subunit behind at the promoter region. The end of a gene is marked by a terminator sequence that forms a hairpin structure in the RNA. The subclass of terminators require a recognition protein known as ROP to function. A process of transcription. In prokaryotes, there are major types of RNAs: mRNA, tRNA, and RNA. All three RNAs are needed to synthesize a protein in a cell. The mRNA provides the template, tRNA brings amino acids and reads the genetic code, and RNAs play a structural and catalytic role during translation. This is single DNA-dependent RNA polymerase that catalyzes transcription of all types of RNA. It binds to the promoter and initiates transcription initiation. The polymerase's binding sites are called promoters. It uses a nucleoside triphosphate as substrate, an energy source, and polymerases in a template dependent fashion following the rule of complementarity. After the initiation of transcription, the polymerase continues to elongate the RNA, adding one nucleotide after another to the growing RNA chain. Only a short strength of RNA remains bound to the enzyme. When the polymerase reaches the terminator at the end of a gene, the nascent RNA falls off, so also the RNA polymerase. The question is how the RNA polymerases were able to catalyze the three steps: initiation, elongation, and termination. The RNA polymerase is only capable of catalyzing the process of elongation. The RNA polymerase associates transiently with the initiation factor sigma and termination factor rho to initiate and terminate the transcription respectively. Association of RNA with these factors instructs the RNA polymerase either to initiate or terminate the process of transcription. In bacteria, since mRNA does not require processing to become active, and also since the transcription and translation take place simultaneously in the same compartment, many times the translation can begin much before the mRNA is fully transcribed. This is because the genetic material is not separated from other cell organelles by a nuclear membrane. Consequently, transcription and translation can be coupled in bacteria. In eukaryotes, there are at least three RNA polymerases in the nucleus. There is a clear division of labor. The RNA polymerase 1 transcribes RNAs 28S, 18S, and 5.8S, whereas RNA polymerase 3 is responsible for transcription of tRNA, 5S rRNA, and snRNA. The RNA polymerase II transcribes precursor of mRNA, the hnRNA, heterogeneous nuclear RNA. In eukaryotes, the monocistronic structural genes have interrupted coding sequences called as exons, express sequences, and non-coding sequences called introns, intervening sequences. The introns are removed by a process called splicing. hnRNA undergoes additional processing called capping and tailing. In capping, an unusual nucleotide methyl guanosine triphosphate is added at the 5' whereas adenylate residues 300 poly A are added at the 3' end tailing. Thereafter, this processed hnRNA is now called mRNA is transported out of the nucleus for translation. The split gene feature of eukaryotic genes is almost entirely absent in eukaryotes. Originally, each exon have been coded for a single polypeptide chain with a specific function. Since exon arrangement and intron removal are flexible, the exon coding for these polypeptide subunits act as domains combining in various ways to form new genes. Single genes can be produced from different functional proteins by arranging their exons in several different ways through the alternate splicing mechanisms, a mechanism known to play an important role in the generation of both protein and functional diversity in animals. Introns would have arisen before or after the evolution of eukaryotic gene. In introns arose late. How did they enter the eukaryotic gene? Introns are mobile DNA sequences. They can splice themselves out as well as into specific target sites, acting like mobile transposon like elements that mediate transfer of genes between organisms. Horizontal horizontal gene transfer occurs between lineages of prokaryotic cells or from prokaryotic to eukaryotic cells and between eukaryotic cells. HTT is now hypothesized to have played a major role in evolution of life on Earth. DNA is the genetic material that carries genetic information from generation to generation. At this stage, an attempt will be made to determine what manner the genetic information occurs in a DNA molecule. The translation of proteins follows the triplet rule. A sequence of three mRNA base codon designates one of the two different kinds of amino acids used in protein synthesis. Genetic code is the sequence relationship between nucleotide in genes mRNA and the nucleic acids in the proteins they encode. There are 64 possible triplets and 61 of them are used to represent amino acids. The remaining three triplet codons are termination signals for polypeptide chains. Since there are only 20 amino acids involved in protein synthesis, most of them are encoded by more than one triplet. Two things make this multiple degenerate coding possible. First, there is more than one tRNA for most amino acids. Each tRNA has a different anticodon. Second, the pairing is highly specific for the first two portions on the codon, permitting Hudson and Crick base pairs A to U and G to C to be formed, but at the third position there is a great field of flexibility to which base pairs are acceptable. Most part of the genetic code is universal, being the same in prokaryotes and eukaryotes. The order of base pairs from a DNA molecule controls the kind and order of amino acids found in the proteins of an organism. This specific order of base pairs is called genetic code. The blueprint establishing the kinds of proteins to be synthesized which make an organism unique. Marshall Nirenberg, Severo Ochoa, Haemoglobin Corona, Francis Crick, and many others have contributed significantly to decipher the genetic code. The order in which the bases are arranged in mRNA dictates the order in which the amino acids are arranged in. The order which the bases are arranged in mRNA, RNA decides the order in which the amino acids are arranged in proteins. The salient features of a genetic code are: genetic codon is a triplet code and 61 codons code for amino acids and 3 codons do not code for any amino acid and functions as stop codon. The genetic code is universal. It means that all known living organisms are nucleic acids and the same 3 base codons, triplet codon, direct the synthesis of proteins from amino acids. For example, mRNA UUU codon codes for phenylalanine in all cells of all organisms. Some exceptions are reported in prokaryotic, mitochondrial, and chloroplast genomes. However, similarities are more common than differences. A non-overlapping codon means that the same letter used for two different codons. For instance, the nucleotide sequence GUC represents two codons. Only two codons. It is a comma-less, which means that the message could be read directly from one end to the other. No punctuation are needed between two codes. A degenerate code means that there are more than one triplet codon and could code for a specific amino acids. For example, codons GU, GUC, and GUA and GUG code for valine. Non-ambiguous code means that one codon will code for one amino acid. The code is always read in a fixed direction, 5' to 3' direction, called polarity. AUG has dual functions. It acts as the initial codon and also codes for the amino acid methionine. Comparative studies of mutations and corresponding alteration in amino acid sequence of specific protein have confirmed the validity of the genetic code. The relationship between genes and DNA are best understood by mutation studies. The simplest type of mutation at the molecular level is a change in nucleotide that substitutes one base for another. Such changes are known as base substitutions, which may occur spontaneously or due to the action of mutagens. A well-studied example is sickle cell anemia, in which humans which results from a point mutation of an allele of B hemoglobin gene BHB. A hemoglobin molecule consists of 4 polypeptide chains, two alpha chains, and 2 beta chains. Each chain has a heme group on its surface. The heme groups are involved in the binding of oxygen. Human blood disease. Sickle cell anemia is due to abnormal hemoglobin. This abnormality in hemoglobin is due to a single base substitution at the sixth codon of the beta globin gene from CAG to GTG in beta chain of the hemoglobin. It results in change of amino acid to glutamic acid to valine at the sixth position of the beta chain. This is the classical high viable hypothesis. It is a hypothesis proposed by Crick 1966, which states that the RNA anticodon has the ability to wobble at its 5' by pairing it even non-complementary base of mRNA codon. According to this hypothesis, in codon anticodon pairing, the third base may be complementary. The third base of the codon is called wobble base, and this position is called wobble position. The actual base pairing occurs for first two positions only. The importance of wobbling hypothesis is that it reduces the number of mRNAs required for polypeptide synthesis and it overcomes the effect of code generation.